Peroxisomes are the organelles where catabolism of very long chain fatty acids like this one begins through the process of beta oxidation. This converts them to medium chain fatty acids by removing pairs of carbons as acetyl coenzyme A, and the medium chain fatty acids are then transported to the mitochondria and completely degraded, which is covered in more detail in another video. If a patient has dysfunctional peroxisomes, what happens? Well, the main problem is that you get accumulation of very long chain fatty acids both in tissues and in the blood, and this can cause congenital disease and also diseases acquired during life. Now, in particular, the main pathology you're going to see with peroxisomal diseases is demyelination. There are three diseases of peroxisomal dysfunction which you should know for your boards: Zellweger syndrome, Refsum disease, and adrenal leukodystrophy. So, what causes these three diseases? Mutations in genes that have to do with peroxisome creation, import of enzymes into peroxisomes, and the functions of the enzymes in the peroxisomes. So now let's go through what these diseases are, what causes them, and what they look like. Zellweger syndrome is the most severe and it presents with significant disease at birth, and these patients typically die within the first year of life. So what genes can be mutated to produce Zellweger syndrome? The most important enzyme families to know about here is the peroxin or PEX genes, and these peroxins are important both for the biogenesis of peroxisomes and for importing enzymes into peroxisomes. Inheriting a defective copy of one of the genes in this family from each parent is the cause of most cases of Zellweger syndrome. So what inheritance pattern is this? That would be autosomal recessive. Now, from birth, the first thing you'll notice in a newborn with Zellweger syndrome are flattened facial features within a broad nasal bridge. Soon after, you may notice evidence of demyelination. So, what kind of symptoms do you see in a baby with demyelination? You can see hypotonia, hearing and vision impairment, and seizures. Last but not least, there may be evidence of liver failure or renal failure. Next, let's go over Refsum disease, which is by a defect in the gene phytonyl coenzyme A hydroxylase, which also has autosomal recessive inheritance. This is a peroxisomal enzyme that normally helps break down a molecule called phytanic acid, which is associated with chlorophyll, and therefore you get it from the diet. If this enzyme is deficient, phytanic acid builds up and causes a variety of symptoms. It first presents in adolescence and mostly starts with neurologic symptoms like worsening of vision, sense of smell, and progressing to hearing loss and ataxia. Years later, you can also see changes in the skin like ichthyosis and cardiac conduction abnormalities. So, how do you treat this? Well, phytanic acid is associated with chlorophyll, so what if you just don't eat vegetables? Well, actually, surprisingly, eating green vegetables doesn't affect these patients very much, but what they do need to avoid in their diet is animal fat, particularly from grazing animals. Cows, for example, eat grass all day, and phytanic acid gets concentrated in their fat way more than you would see in any vegetable. So, this is what patients with Refsum disease need to avoid. Last but not least is adrenoleukodystrophy. The gene for this one has a great name, very easy to remember. It's called ABCD1. This encodes a protein that forms part of a channel which is used to import very long chain fatty acids into the peroxisome. So without it, peroxisomal functions won't be carried out properly. Now ABCD1 is on the X chromosome, so this one is an X-linked recessive disorder. So who do you think is more likely to get this, males or females? That would be males, since they only have one X chromosome, so they only need to inherit one defective gene, which is often from their mother, who is an asymptomatic carrier. So what's the classical clinical presentation? That one's a trick question because this one comes in a few different flavors. The most common three types by far are the childhood cerebroform, the adrenomyeloneuropathy form, and the adrenal insufficiency only form. The childhood cerebroform accounts for 35% of adrenoleukodystrophy cases, and it presents in early childhood as progressive neurodegeneration. Although in the very beginning of the disease, the child may just have hyperactivity and disruptive behavior at school, so it's often first misdiagnosed as ADHD before it worsens. This will be fatal in a few years if untreated. The adrenomyeloneuropathy form accounts for about 45% of cases, and this presents in young adults like in the 20s and 30s with progressive neuropathy and weakening of the lower extremity. These patients will also develop adrenal insufficiency, and about half of them may eventually develop cerebral symptoms such as cognitive decline and behavioral change. The last form accounts for 10% of cases and usually occurs in early childhood with isolated renal insufficiency, but decades later can progress to have demyelinating disease as well. The proteasome is a large complex of proteins whose function is to degrade other proteins into small fragments about 8 amino acids long. This is a very important function since misfolded or damaged proteins can cause disease, such as the beta amyloid proteins in Alzheimer's disease. Proteasomes look kind of like a barrel and have a regulatory component shown in red and a catalytic part shown in blue. Since you don't want these things going around degrading all your protein, cells have a system that allows the proteasome to be selective. So here's a protein that in some cases we might not want to have around, so we want to degrade it. This is the protein hunting which aggregates and causes Huntington's disease. Proteins that need to be degraded are recognized and then modified by enzymes called ubiquitin ligases, which covalently attach a small protein called ubiquitin to the target protein. Ubiquitin binds to the proteasome's cap, which causes the barrel to open and allow the protein inside where it is degraded.
Let's move on to the cytoskeleton. Do you remember the cytoskeletal elements? We've got microtubules, actin, and intermediate filaments. We call it the cytoskeleton, but it's not just to hold the structure of the cell. It also has roles in transporting things around the cell, along microtubules helping the cell move around using flagella, and even helping the whole body move, since muscle contraction depends on actin and myosin. You can get a sense of how the cytoskeleton is organized by this picture, where the nuclei are blue, the microtubules are green, and actin is red. Actin and myosin compose microfilaments and are the thinnest structure in the cytoskeleton, with a diameter of just 7 nanometers. You probably know that actin and myosin work together in muscle contraction, and that's covered in a different lecture, but can you think of anything else that actin does? It's an important structural component in microvilli. Do you remember where you might find lots of microvilli? One example is the brush border of the small intestine, where microvilli are used to increase surface area and therefore increase absorption. Remember, a villus has a lot of cells and each cell has lots of microvilli. So microvilli are a lot smaller than a villus. Actin is also used in adherence junctions, which are common in epithelial and endothelial cells, and are used to hold cells together. Intermediate filaments are generally involved in cell structure and have an intermediate diameter of about 10 nanometers. Some examples of intermediate filaments are, but I'll come back to these in a couple minutes. And microtubules are the largest part of the cytoskeleton with a diameter of about 25 nanometers, and they're generally involved in movement of things within the cell, or of the cells themselves. We see them in cilia and flagella, and also in the mitotic spindle and centrioles. Okay, let's talk more about microtubules. What color are they in this image? They're the green ones. They're about 24 nanometers in diameter, which makes them the largest cytoskeletal element. They're cylindrical structures formed by polymerized dimers of alpha and beta tubulin. Each dimer has 2 GTP bound to it. Also remember that they have a positive and a negative end. They grow together in chains, maintaining their polarity. While their growth is relatively slow, they can collapse quickly when needed. So what are microtubules used for? Well, there's really a lot of things. Basically, anytime you want to move something large around a cell, you need to use microtubules. For example, during cell division, what do you want to move around? This would be chromosomes, since you have to separate homologous chromosomes so each cell ends up with a copy. So microtubules, which are these green lines, are anchored to centromeres and pull them apart. What else can we move around? How about carrying vesicles filled with proteins along an axon of a neuron? Now, as I mentioned before, there's a positive and a negative end, and you need a different motor protein depending which direction you're going. If you want to carry something towards the positive end, which is called anterograde transport, you can attach your cargo to a motor protein called kinesin. And if you're going towards the negative end, which is retrograde transport, you can use the motor protein dyne. In neurons, the positive end of the microtubule is at the distal end of the axon, whereas the minus end is near the cell body. So which motor protein is used to move something towards a synapse? That would be kinesin. And by the way, if you're moving a vesicle filled with proteins along the axon using kinesin, Where were these proteins synthesized? If they're in a vesicle, then they were made in the endoplasmic reticulum, not in a free ribosome. And remember, in neurons, the rough ER is called the nasal body. The last function of microtubules I want to cover is their role in cilia and flagella, but I'll save that for the next slide. First, let's go over some things that interfere with Alright, so why would you want to interfere with microtubules? Well, like I said earlier, one of their functions is in cell division.
So anytime you want to block cell division, you can use a microtubule inhibitor. Some examples of microtubule inhibitors are drugs that treat cancer, gout, and parasitic infections. Cancer should be obvious here since cancer cells divide a lot, but since immune system cells also require lots of cell division, microtubule inhibitors have kind of an immune suppressing effect too, which is why they can treat gout. And for the parasites and fungi, you want to use inhibitors that are specific to those types of species and have minimal effect on human microtubules. Some examples that are most likely to show up in step 1 are medbendazole, which treats helminths, crucifulvin, which treats fungi, colchicine, which is a super important drug which treats gout, and vincristine, vinblastine, and paclitaxel, which are all chemotherapy drugs. Now, why does colchicine treat gout? Well, first of all, what is gout? Basically, if you have high levels of uric acid in your blood, it will crystallize and these uric acid crystals will deposit in joints, especially the big toe. This ultimately causes recurrent acute inflammation. Alright, so what drug can you use to treat patients long-term for gout? That would be allopurinol because allopurinol prevents uric acid formation. But during an acute attack, what do you use? So that would be colchicine, which doesn't affect uric acid directly, but is basically working as an anti-inflammatory drug, and you could also use NSAIDs or ICE. Now for these chemo drugs, you learn more about these in the Hem/Onc chapter, but what do you think might be an important adverse effect of inhibiting microtubules with vincristine, vinblastine or paclitaxel? These drugs can cause bone marrow suppression since again we're preventing cell division, and like it says in your book, you can remember these with a mnemonic: microtubules get constructed very poorly. Alright, so what happens if microtubules aren't working properly? Well, it depends on the specific problem, but one example is called Chidiac-Higashi syndrome. This is usually caused by a mutation in the lysosomal trafficking regulator gene or LYST, which normally is required for organizing proteins into mature endosomes for microtubule-dependent transport. Since granulocytes need this to make their granules, this defect makes patients susceptible to recurrent pyogenic infections. Since macrophages and neutrophils can't make the weapons they need to destroy bacteria. These patients also have albinism since melanin can't be transported out of melanocytes, and peripheral neuropathy since vital proteins can't be transported to the ends of long peripheral neurons. Before we move on, let's see if you remember which direction these proteins. Dynein is used for blank transport and kinesin is used for blank transport. The options here, by the way, are either anterograde or retrograde. The answer is retrograde or towards the minus end for dynein, and anterograde or towards the positive end for kinesin. So as I mentioned a minute ago, another role of microtubules is in cilia and flagella. Both cilia and flagella are made from microtubules arranged in a 9 dimers plus 2 singlets formation. So here I'm showing you an electron micrograph of a cross section through cilia, and you can see the 9 dimers around the periphery and the 2 singlets in the middle. Now in the last slide I mentioned that dynein is used for retrograde transport along microtubule. Dynein can also be used to bind microtubules for a different purpose, which is to link these doublets together. Then ATP can be used to cause them to slide along each other, which makes the cilium. Flagella and cilia have a similar structure if you take a look at the cross section, but they also have some functional differences. First of all, flagella move in this kind of propeller pattern since they're trying to generate force that pushes the cell away from where it's pointing. Cilia beat back and forth since they're trying to keep the fluid outside the cell moving in a consistent direction. Flagella are a lot more common in single-cell organisms like bacteria, but can you think of any human cells that have flagella? Sperm are the first ones that come to mind for me. How about cells that have cilia? Two important ones are cells in the lung that keep mucus moving out of the lungs, and cells in the fallopian tubes which move the egg towards the uterus. Okay, so what happens if you have an autosomal recessive defect in dynein? It's called cartogenous syndrome or primary ciliary dyskinesia, and if you have it, your cilia and flagella won't be able to move at all, so all these functions I just mentioned won't work.
Both males and females will be infertile since sperm can't move and the egg won't be pushed along the fallopian tube to the uterus. Patients with carcinogenesis also commonly have bronchiectasis and recurrent sinusitis since cilia are required to sweep particles out of the lung. And lastly, it's associated with situs inversus, which is a condition in which major organs are found in the lungs but not found in the opposite side of the body. For example, the liver should be found on the right side, but here it's found on the left.